Okay, so first of all, I'd like to thank the organizers for giving me this opportunity to be here. As Rafael said, I'm a RISE alumni. And I have my PhD co-advisor, Lydia Kabraki, and my longtime collaborator as well in the audience. So it's great to be here. Every time I come back, I see these new institutes, and they are growing bigger. So it's really exciting that you have this opportunity that under this umbrella of this initiative, because right now I feel like a crazy moment, more of a revolutionary kind of moment in AI for ethics as well as in biology. So in my talk today, I'll be taking you through some of our work on foundation models for advancing biologics design, more specifically protein design. In my team, as Rafael mentioned, we do work on trustworthy aspect of AI models, LLMs, as well as new architectures. And I'll be happy to talk about all of those efforts in a private conversation. So why we worry even after AlphaFold moment for AI for protein modeling? There are several challenges because as we know that even though with all of the large data sources available today, function annotated data is still very limited. The protein sequence data is available more than the resolved structure. When I mean resolved structure, I mean experimentally resolved structures. And in many downstream events like binding or function, it is the dynamics of the protein that actually govern the outcome. Now, the proteins being implicated as many disease targets, and also they can be used as for therapy, for treatment, it's very important that we understand the full landscape of these systems from sequence all the way to dynamics. And what I want to cover today to show you that how some of the foundation models, some of that do share an architecture similar to a language model, but some of them are multimodal intrinsically, as we know that from protein sequence to dynamics, it actually goes from 1D to 3D, and then the time aspect, the temporal aspect of it, which you can consider as the 4D. So I want to show you, some examples that where these models trained on broad sequence and or structural data set can be used for enhanced modeling of such systems. Now, a disclaimer, I come from a tech industry lab, not from a pharma or from a biotech, so it's very difficult for us to test our predictions in wet lab. And we consider ourselves extremely lucky if we do have such an option where we can test our design for their efficacy, for their activity in wet lab. Even though we do so either we go and test our predictions on open source public benchmark, or we do have actually engagement with different pharmaceutical or biotech clients with whom we can test the predictions. But unfortunately, because of proprietary issues or client privacy, we cannot publish those results. So, but few years back, we were lucky. This was during end of COVID, around that time, where we were able to do some modeling of antimicrobial peptides as well as of small molecules, and we were able to test some of these designs in wet lab. And what we did found that in both of these cases, in the first case, our goal was to design using generative AI, new antimicrobial peptide that simultaneously satisfy different aspects like broad spectrum potency, low toxicity, and low resistance onset. We were able to validate them in the wet lab, and what you see on the screen here are two new peptides that we found. They were not reported in large databases or even patent databases. At the same time, around the same time, we also collaborated with experimental labs in University of Oxford and Diamond, where we were able to use generative AI to design small molecules specific to protein targets. These targets were from SARS-CoV-2, so MPRO and spike protein. And we found that these targets, that the small molecules that we used, that we designed using our generative AI approaches, indeed show inhibition of these SARS-CoV-2 targets. And overall, what we do see that in both of these cases, one being peptide design, one being small molecule inhibitor design, in both cases we found, and this was validated through our experimental collaborator as well, that we do see higher success rate with generative AI, and also the time to discover and confirm actually is much faster. But for today's talk, I'm not going to do a deep dive of those prior work. But what I want to show you is some new approaches that we are coming up with and looking into, which also tackles some of the very difficult aspect of modeling protein system. The first one is learning from the low data regime, which we all know, like anytime a model, a pre-trained model looks good because it has been trained from million to billion scale data. But actually, when we are working with pharma or with biotech, they come with 100 to 1,000, sometimes function annotated data point. So this is like a new approach that we came up with, which we name as representation reprogramming, to look into prediction and design tasks by learning from small amount of data. In the second part of the talk, I will go beyond. The first part of the talk only focuses on learning from sequences alone. In the second part, I will be talking about learning from multimodality, to be precise, sequence and structure. So I'll talk about a geometric protein encoder training and also doing inverse folding with structural consistency. And in the last part of the talk, which is very interesting to me at least, where we are trying to model dynamics that is driven by underlying physics of protein systems into machine learning or deep learning model training. So that's like the structure of today's talk. So first I'll start with representation reprogramming. So what do I mean by representation reprogramming? As you know, like when we are talking about these foundation models, specifically they are like when it comes to learning on text, they are specifically mostly transformer based. However, they are data hungry, right? So we need a lot of data points to train them. But like I mentioned, for many protein property prediction tasks, the amount of annotated data could be very limited. So what we do here, we basically learn how to reprogram an already learned English language model. It could be any other natural language model, but let's say English for this case. And then we learn how to reprogram that or repurpose that to predict properties of protein sequences. And the reason we go this approach is that if you look into literature, there is already some known linguistic similarity shared between both protein language or any biological language for say, as well as and natural language. So we build on top of that, and what we do is we take, let's say, an off-the-shelf English language encoder like a English BART, and then we extract the embeddings of English words from it. So let's say that there are 30,000 or English vocabulary that the model has seen, so it will give us 30,000 vocabulary embedding, word embedding. What we then learn is a new mapping function that will map this English word embedding to protein amino acid embedding. So this function is called theta, that let's say we are calling it theta. So if we are called this approach R2DL, that representation reprogramming through dictionary learning, because what we want that this target embedding or the amino acid embedding to be mapped as sparse linear mapping of the original English word embedding. So we use a KSVD approach for that. And then we also have a task at hand. Let's say that the task is to predict that if the protein adopts an alpha helical structure or a beta sheet, right? So it's like one or like it's two-class problem in this case. So using this task-specific loss, we update this parameter that we are learning, the mapping function we are learning theta, such that we can reprogram the original English BART model to do this protein property prediction task. So we are not training the original network, which could be million to billions of parameters. Rather, we are only training this mapping function, which is much smaller number of learning parameters. So it's much cheaper to train, and it's also because it's cheaper to train in terms of data as well as in terms of compute. And here I do show you some result. So I don't know how much you can read the font. I don't know if it's too small or not. But what you are seeing here is two different plots, one for accuracy and one for efficiency. And for accuracy, for each, we have three different architecture or approach. The blue one, R2DL, or the representation reprogramming. The second one is training from scratch. So it's a supervised learning approach where we are training a whole deep neural network BART model on this set of protein sequences. And the last one is pre-training, where we are using a protein language model like ESM or something that is available off the shelf. And then we are subjecting it to different tasks like predicting toxicity, secondary structure, antibody affinity, etc. And on the right, I show the same benchmarks, same property prediction benchmarks and the same set of approaches, but the y-axis here is data efficiency. So overall, what we find that the R2DL approach it actually provides almost equal or sometimes even slightly better accuracy on all of these different property prediction tasks, while being 10 to the power 5 times more data efficient in some of the cases. So this is very interesting. This kind of opens the door that if we can subject some of these large architectures to learning from small data, and I don't know if that slide is, yeah, I have a slide in the middle that's actually now hidden, where we show that if we actually do some fine-tuning, partial fine-tuning of, let's say, a protein BERT architecture or just add a linear head, they actually lose accuracy. And this is a known problem that partial fine-tuning or learning a linear head on top of a deep learning architecture may not be the most efficient thing, particularly when we are subjecting them to learn from small amount of data. And when we compare R2DL with those approaches, we do see that our approach actually shows better accuracy across these different tasks. Now, that is about property prediction. Is this of any use, this model reprogramming for design task? In fact, it can. So this is one example where our task here is to design the CDR loops of antibodies. If you are familiar, the CDR loops of the antibodies are actually the functionally implicated region, they are loop. And the rest of the antibody sequence are pretty much the same. So people, the antibody scientists, they actually look for optimizing these CDR loops to design new antibody. So the way we cast this problem is that we cast it as a slot infilling. So we have a long string of amino acid here, as you can see, and the CDR loops are the one where I have star. So they are actually masked during this treatment, the model learning problem. So the way we ask the model to learn that, okay, we have masked the CDR position, and through training you need to learn how to predict accurately those masked position. But again, we want to go back to model reprogramming, because in these cases, again, for antibody optimization and stuff like that, you could end up with very limited data set. So what we do, again, we take an English BERT encoder, and we cast this as a slot infilling problem through masked language model objective. And we learn, in this case, 2 linear projection layers. One that takes the original amino acid sequence and ends up with embedding that actually can be fed to the English language encoder. And then another encoding, which will take another linear projection layer that will take output of this English language encoder or its prediction and will map them back to the amino acid embedding space. So we are learning 2 basically projection layers. One we call, I think, theta, and one we call gamma. So it's, again, we are casting it not just input-output translation, it's also we are casting it as a domain translation problem. And by learning only this linear projection layer, again, which is much, much smaller in terms of number of parameters than this English encoder model, we are being able to successfully design, successfully solve this CDR infilling problem. And when we are looking at how this approach does, so here we are looking at different metrics, perplexity, RMSD, TM, which are structural consistency metrics, amino acid recovery, how many we are recovering compared to the ground truth protein, and diversity. And here we have some baselines like ABLANG, which is a language model trained on antibody sequences. It was trained, I think, on million scale sequences, where we are training ours on few thousand only. And ProtBERT, again, another approach such as, and the original English BERT model. So you could see that ProtBERT and English BERT, while they are high on recovery, they are literally, there is almost very minimal diversity in terms of CDR sequences, whereas we know that CDR sequences actually, if you look in nature, they are more sequentially diverse. And in our case, the reprogramming BERT case, we find that we are good on the recovery side, and while we are getting also higher diversity. So it seems like this kind of approach, particularly when being subjected to learning from small amount of data, can give us benefit in terms of designing novel sequences that are consistent, both sequentially and structurally, and also diverse. Okay, so now we are moving to the second part of the talk, where we will move away from learning only from sequences, and we'll start focusing on how we can learn joint representation, which combines information from sequence as well as from structure. So first I'll be talking about something called a protein geometric encoder, or we call it GearNet. So GearNet, where we build a multi-relational graphs for protein. The nodes here are situated on the C alpha atoms, and then we are looking into different kind of age types. So the ages here are defined not just by looking into the sequential relationship, but also within a epsilon neighborhood in 3D space, how many neighbors it is interacting with. So that's sort of how we build the graph, which encodes both sequential and spatial information. And then we do typical relational message passing over these nodes. So the node now have not just the sequence features, they also have some spatial features because we are building the graph as such. And then we do relational message passing over that. However, when we are subjecting this resulting architecture, which is a graph neural network, message passing graph neural network, we do see just message passing over the node level representation is not enough. And you need to account for the edges. You need to model the edges. So what we do, and if you look into AlphaFold 2, it actually does something very similar. They do a triangle update to end up with pair representation. However, it's more expensive because if your sequence is n amino acid long, you are doing n square, because you are looking at all pair. Rather, what we do here, we only look at actual edges, and then we build a graph on the edges, so where the now edge are being the node itself, and then we do an edge message passing. So we are doing two levels of message passing, one over the nodes and one over the edges, and we are updating both node and edge features as such. As a result, it's like the resulting architecture we call GearNet, and we do have looked into both options where we do have edge message passing and where we don't have edge message passing. So we call it GearNet or GearNet-Edge. And what we find that GearNet-Edge version actually does much better than the original GearNet. But GearNet also does much better than unsupervised protein property prediction task like enzyme commission prediction task or fold classification task, much better than existing transformer-based architecture or different graph architectures. So it seems like this learning to model the relation between different amino acid across sequence and spatial features do have benefit. But when we are focusing on learning to update the edge features, that actually even provide us better benefit because we do see the accuracy or the correlation in across all of the tasks go high when we switch from GearNet to GearNet-Edge. So for GearNet is mostly for supervised settings. So everything you see here, we are training the model on the property prediction dataset itself and checking how good it is compared to the other approaches which are graph-based or LSTM-based or transformer-based. In terms of expanding that to foundational model perspective, we also introduced geometric pre-training. And where in geometric pre-training, we take two different approaches. The first one is where we do multi-view contrastive learning. So basically, we take, let's say, if we are focusing on a part of the protein, what we do there is like we create different correlated view by, let's say, do subsequence cropping, by masking a random edge within, that small part of the protein. We can also do subspace cropping, or we can ask the model to predict, identity. So by creating these different views, now the task of the model is to ensure that the correlated views are more similar compared to the non-correlated view. So this could be one way to introduce geometric pre-training on large-scale data where we do not have access to the final annotation, but we are just learning from the sequence and structures. The other way we can do it is that we can ask the model to do self-prediction. So again, if you are familiar with language model, the mass prediction is, mass language modeling is kind of a self-prediction task. And document similarity learning is like the multi-view contrastive learning sort of task. So the other kind of pre-training, we ask the model to predict its residue types or of the masked amino acid or the distance with its neighbors and so on and so forth. And when the one thing I wanted to just mention that in each of these cases, we are training with the downloaded structure from AlphaFold DB version 1 and version 2, which is less than 1 million structure. That was, at that time, that was the largest amount of structural information available on protein monomer systems. And we compared these architectures with pre-trained sequence model that are trained on 10 million to 2 billion sequences like ESM, LMGVP, ProtBART, etc. And when you look at this chart, overall what we see that across, let's say, enzyme commission and gene ontology prediction, we do see that when we are using GearNet Edge or with multi-view contrastive learning or differential prediction task, the model actually performs better than the protein language models which were trained with 10 million to 2 billion sequences. So it seems like, again, of course, you can argue that, okay, here we had to have the structural information available, but on the benefit side of it, we are learning, only on less than 1 million data points compared to 10 million to 2 billion data points. So far, this is learning from sequence and the structure, the coordinate information alone. But those of you working with protein system or any biological system, the surface features can be also meaningful. So we took a step further and we started integrating the features of the amino acid extracted from a protein language model like ESM with structural features or geometric features, but also surface level features. And the surface level features could be computed with something called D-massive. And when we integrate all of these three, we do see that in tasks like zero-shot fitness prediction, where the protein is being mutated at a point or two or a few, the task is to predict this new mutational variant, how fit this one is with compared to the wild type. And this is a known task. It's a very important task because when we are designing new protein mutant, let's say to optimize its binding affinity, one preliminary aspect is to check that it is going to be fit for the system, the host system. So, I'm not going to show you bore with showing you tables of with the different benchmark and different baselines anymore. But what I want to show you that compared to language only model, which is ESM here, what we are trying to look in this figure is that how good our S2F, where we are only integrating or combining sequence features with structure features, and S3, meaning sequence, structure, and surface, how the learning from both sequence and structure and sequence, structure, and surface level features are more important than learning from language only or the sequence only features. And what you see here is the correlation with experimentally estimated values of protein fitness after there are mutation in these sites, in these amino acid sites, correlation of that with the model predicted values. And red is higher correlation, higher Spearman correlation, and blue is low. So as you see, particularly when we are looking at this correlation where these residues that are mutated are quite distant apart in the sequence, the ESM model actually predicts the correlation with the ESM predictions are quite poor. However, as we are starting to integrate the structure feature and more so the surface level features, now we see that these epistatic effects in this particular protein, the IgG binding domain of protein G, are modeled much, much better compared to learning from sequence only features alone, which shows that it is important not just for property predictions like enzyme commission or fold classification, but also for fitness prediction upon mutation, learning from sequence and structure is very important. Okay. Okay, so now, and for this part, I will also show you another example that where we need to learn from both sequence and structure jointly. But this time it's not for a prediction task. This time it's going to be a design task. And this design task is called inverse folding. So for folding, you have protein sequence, you predict the structure. For inverse folding, you have a backbone structure as an input. You need to predict the sequence corresponding to it. These models typically are trained with cross entropy loss. That architecture could be anything, but the cross entropy loss between the ground truth sequence and the predicted sequence. However, even the best inverse folding model, and there are quite a few ESM fold, GVP, protein MPNN, which were published a few years back, those models, there seems to be quite some room for improvement. If during training, these models can be, can receive some structural consistency feedback on the sequences they are predicting during training. And so you can think, okay, I have now AlphaFold or Boltz2 or ESM fold. Why can't I use them during training? And the problem here is twofold. That first, even if you call AlphaFold, like I'm showing here you a chart that how much single inference call, how long it takes. That is typically like, I would say, 10 to 35 or so second. I don't know where Boltz 2 stand in here. That time it was not available. But whatever, you can think that during an actual training of a deep learning model, which is being trained for this inverse modeling task, it is not feasible to call those structure predictor model again and again and again. And the other thing is that those models are not also easy to differentiate through. They are not, you cannot do gradient backpropagation easily through them. So what we do is a very simple idea that where we try to distill the prediction from AlphaFold in a much smaller model, which we call as alpha distill. Basically, our goal here is to distill the predictions of AlphaFold in a much smaller network. So it's going to be, at best, it can be as good as AlphaFold. It cannot go beyond that. But however, it's going to be much cheaper. And because it's like, like a BART-like architecture, we can easily update our inverse folding model using prediction from it. So we take the predictions from AlphaFold, the predicted TM, PTM, and the predicted PLDDT. That's our output value. And we train a small BART-like model on AlphaFold on the original sequences that are predicted to learn the AlphaFold predictions, confidence values on those sequences. And this is the one we call alpha distill. Once we have alpha distill available, we bring it to the loop of this inverse folding model training. So now, during this inverse folding model training, the model is not just going to be updated from the gradient of the cross entropy loss. For each predicted protein sequence, we are going to pass it through this AlphaFold distill model, which is cheaper, and get the predicted PTM or PLDDT values. And then we are going to use that to estimate the structural consistency score with the original sequence. And so the combination of the C loss and the structural consistency loss is our final loss. So that's how we train the inverse folding model. When we subject this inverse folding model to protein design task and protein infilling task, we do see that actually this can be used to enhance the existing inverse folding model like GPP, protein MPNN, and PyFold all across. So it's like a model agnostic approach. And also what we do see that when we are adding the structural consistency loss in inverse folding model training, the generated sequences show more higher consistency with the ground truth as well as showing more diversity, which is again important for this design task. Okay, I hope I'll be on time. I think 5 minutes? Yeah, good. Okay. So now the last part of the talk where we have covered how to learn from sequence to some aspect, how to learn from sequence and then structure and then also from surface level features of the protein. How about dynamics? We, those who work with, all of us know biology is nothing without dynamics. Now, and this is actually, I want to emphasize, this is actually a very new area of research, and only few, I would say, over the last 2 years have been looking into it, and I believe a lot can be done here. Now, typically how we learn the protein dynamics, we have the physics-based molecular dynamics simulation engine. There have been a lot of work going on to advance those physics-based function learning so that the simulation can be done with those MD simulator. However, they are more expensive in terms of both compute and time. And as we all know, AlphaFold gives us one structure at the end for each sequence, so it's missing the dynamic aspect part of it. So the idea we had here that, can we, let's say, make AlphaFold more aligned with the underlying energy landscape? Can we do that? And to do so, we assume that we have the trajectory from the molecular dynamics of some proteins available. So we are training them. We can fine-tune the AlphaFold further on this molecular dynamics training set. However, MIT lab, one lab from MIT, they actually, there was some result that we looked into. It seemed like, again, there is quite some progress could be done, because one aspect that that often is forgotten, I mean, not who works with physics-based analysis, but for one who works with machine learning models, that there is a Boltzmann probability that determines how this protein landscape would look like. And that's something we wanted to model in our training approach. So we borrowed the idea from something called reinforcement learning from human feedback, if you are familiar, or RLHF, which basically is like some sort of further fine-tuning of a pre-trained model, where the model is being fine-tuned to generate preferred instances over not so preferred ones. And in this specific case, the preferred ones is actually which has lower energy compared to the ones that have higher energy, because that's how the physics-based function determines like which conformation is going to be more preferable at a certain temperature versus which one is not. So we look into the, so assuming that we have the access to the MD trajectory, what we do, we estimate the Boltzmann factor for each pair of trajectory, or for some pairs of conformation. And this Boltzmann factor can be written as such. So xi is one conformation and xj is one. So if we are trying to estimate that if probability of xi is over probability of xj, so this is the Boltzmann factor that exponential beta is in 1 over t, energy of the conformation j minus conformation I. So we can introduce this energy, introduce this factor as we are fine-tuning, let's say, the original AlphaFold model, such that it just does not predict any structure, but now it starts to predict energy-aligned structure. So in practice, what we do, we take the structure prediction module from AlphaFold 3. So up to this, we are not doing much. All of this is done within AlphaFold 3. And now, when we are fine-tuning the AlphaFold 3 structure prediction model, which is a diffusion, all-atom diffusion model, what we are doing, we are augmenting the original loss. So this is the L total is the original loss, which is basically deviation of the predicted structure from the ground truth. We are taking k samples from a particular trajectory, and then we are reweighting the probability of those trajectory with their Boltzmann factor, which is Wxi, and the loss here looks like this. So basically, now we are looking into, if you are familiar again with statistical mechanics, this looks like pi log pi sort of thing, right? So this is like Boltzmann reweighted way of training the loss. And so we call this energy-based alignment, which is basically an enhanced version of direct preference alignment objective, or DPO, and which actually accounts for the energy difference between multiple conformational states, which is weighed further by their physical energies. And I'll skip this for the sake of time, and I just wanted to show you this one result where we are looking into some actual prediction from our approach, and we are comparing that with predictions from the original AlphaFold pre-trained module. And the MD ensemble is here. So the MD, and when we are looking at C alpha root mean square fluctuation, you could see that the blue is the MD ensemble, so it definitely has some peaks. When we are using the pre-trained AlphaFold model, it, besides the N-terminal, actually does not model the flexibility or the conformational fluctuation of the protein at all. However, when we are doing the energy-based alignment, as I presented to you, now we do see that the RMSF or the C alpha flexibility profile, dynamics profile, is much better matched with the ground truth. So with that, I'll close my talk by saying that there are different ways to take foundation models to work with different challenges that we often face during biologics discovery problem. Learning with low data scenario can be addressed by looking into repurposing existing representation from another domain. Definitely structure is important, and regularizing and modeling on structure gives us access to better prediction and generation tasks, performance on those. And finally, we should never forget the underlying physics of this system. So it is very, very important, not just for proteins, but for biologics and beyond, aligning these machine learning models with physics, which is going to provide us better, more realistic picture of this complicated system and can further aid in better biologics design. I'd like to thank you all for your attention, and also thank my collaborators, particularly Ria Vinod, who did the reprogramming work, Igor Melnik for Alpha Distill, Pinghu Chen, Vigil Orelli, my collaborators from IBM, and my collaborators from Mila, particularly Jubai Zhang and Jiarui Lu, for driving the energy-based alignment and gear network. Thank you.
We should have time for a couple questions.
So, in the conformational ensemble calculations, did you have a chance to validate the ensembles against NMR structures for which you have such local sampling is experimentally available?
We actually, so that's a very good question. So we looked into a standard benchmark called Atlas And here again, so this is going to be, because we are training the model on MD ensemble, right? The model at best is going to be as good as molecular dynamics, right? So if the molecular dynamics is missing to model, predict dynamics in some particular region, the model is not going to pick it up, right? So we compared with the MD ground truth ensemble across a number of protein, different families, and we looked into different characteristics like, weak contact, transient contact, are those being correctly captured in our model versus, in the ground truth molecular dynamics ensemble. But you brought up a good point, I look like, like if it can go beyond that. But I'm assuming that, it would be good not just to restrict the training of these models from the molecular dynamics ensemble, but also start to bring in NMR and additional spectrometry data in there.
Just to follow up on
Yeah.
that, you may also want to check training on normal mode calculated.
Yes. Yeah.
Way faster.
Oh, fast. Yeah. True. Yeah. Yes. Yeah. Great suggestion.
Hi, Payel. First of all, great talk. Yeah. So I was asking, like, you talk about small data, training on small data, like you probably trained on AlphaFold DB. Is it like only sequence that you are focusing on, or is it like also the, conservation or relationships from evolution, like from MSA? Because we know about like MSA subsampling, clustering, generating such ensembles like MD. So do you have any comment on that?
Yeah. I don't know if so here, okay, so I skipped it because of time. Thanks for asking the question. So AlphaFlow MD, which where the AlphaFold was further trained with a flow-based objective on a molecular dynamics training set, the same molecular dynamics training set as ours, right? That's what you see the results across different, property like, flexibility at per target, right, or ensemble observables, etc. And then you also see how much you can enhance those results with different MSA subsampling, right? And this is where, ours stand. And MDGen is where they repurpose the GPT model for predicting next structure. And so you see that, even with MSA, right, it's not looking so good, particularly when we are looking at, let's say, pairwise RMSD and stuff like that. So it definitely, the MSA by modeling evolutionary relationship is important, but it cannot compensate for learning from the energy-based relationship across different structures. Yeah.
We have one last question, and then we'll move on.
Hi, great talk. I just quickly want to ask, for the ensemble generation, you mentioned that you used AlphaFold's MSA representation and probably the pair representation as well. I just wanted to ask, did you use the structure module at all for the diffusion step?
Yes.
Like the invariant point attention, et cetera?
Yes, yeah. So, yeah. So if we go back, so we are, okay, so this is like, give me 1 second. Yeah, so this is how it looks like, right? So we are first, using with MSA, we are using the residue level representation, which now is being as a condition on the diffusion module, which actually predicts the structure. And in this case, it's kind of different from the original AlphaFold 3. It's AlphaFlow, because here it's through diffusion, right? It's trained with the flow objective, I believe. But anyway, so as you can see, so we are using the sequence and residue level representation, and then conditioning the diffusion model, which is the structure prediction module here. But further, so this is the pre-training part. So this is like the AlphaFold 3 part. What we are doing, you can consider it as a further tuning step, where we are repurposing the original loss with this energy reweighted loss.
Okay.
To ensure that the model just does not predict any structure. It has now a preference to predict low energy structure for the same sequence.
Gotcha. Okay, thank you.
Okay. Thank you.